there are four basic points that I would like to get across today. The first is that there are really some very significant knowledge gaps about the control of large-scale anatomical homeostasis. And this is roughly, it encompasses our understanding of the relationship between the genome and the anatomy. And I'll show you why things are quite puzzling there. And I will claim that fundamental advances in new biomedicine are going to require not only understanding the molecular mechanisms that are required for this process to happen, but actually also the decision-making by cellular collectives that is sufficient for it to happen. And that a key medium for computation in living tissue is something that is called non-neural bioelectricity. We've developed some techniques to manipulate this layer of basically physiological software, and I'll show you why I say that. And fundamentally, looking towards the future, I will argue that cracking this bioelectric code that will enable a kind of novel approach of using electroceuticals for applications in birth defects, regenerative medicine, cancer, and synthetic bioengineering. And boiling down the whole talk into basically two sentences, what I'm going to tell you is that, like the brain, your body tissues form electrical networks that make decisions. These are decisions about dynamic anatomy, and that we now have the ability to target the system to control large-scale editing that can even override all kinds of genomic default states, with lots of advantages and opportunities for regenerative medicine and synthetic bioengineering. And I show you one of our five-legged frogs to just point out that you're going to see all kinds of weird creatures today. None of this is Photoshop. These are all actual living things that represent our attempt to test some of the models that we have. So let's look at these knowledge gaps. If we think about what is the end game of our field, where are we going? At what point can we all go home? What we would like to have is something that we call the anatomical compiler. The deal is that you ought to be able to sit down and draw the animal or plant that you would like to have at the level of the anatomy. So not at the level of pathways, but at the level of final anatomy, the way that we do with machine parts and things like this. Then you would be able to draw this three-headed worm. And if we knew what we were doing, we would have the capability of having the software which would then compile this anatomical description down into a set of stimuli that would have to be given to cells that would cause them to produce whatever it was that you just drew. So here's this three-headed flatworm. Now, the reason that this is fundamentally important is that almost all of the problems of biomedicine, so except for infectious disease, pretty much everything else, birth defects, traumatic injury, cancer, aging, all of these things would be solved if we had the ability to tell cells what to build. If we had the knowledge to control what it is that cellular collectives cooperate towards building. So all of these things are in some important sense problems of information processing. How is it that cells work together to build these things? And so, of course, we are very far from having anything like this. And only in a few very special cases do we know anything about how to make specific shapes come out. So let's think about why that is. So where is anatomy specified? I mean, we all start life roughly like this. So this is a collection of embryonic blastomeres. And then shortly thereafter, you get something like this. This is a cross section through a human torso. So look at this incredible invariant order. All of these organs are the correct shape, size, position, orientation. Everything is next to the right stuff. It's absolutely staggering amount of order. And so we would like to know, first of all, where is this information coming from? We're tempted to say DNA, but of course we know what genomes specify. The DNA specifies proteins. There's nothing directly in there that specifies any of this. So we would like to know, how is it that cells use the genomically specified hardware that they have to know what to make and when to stop? In regenerative medicine, we would like to know if a piece of this is missing, how do we get the cells to rebuild it? And as engineers, we ask, what's actually possible to build given any particular default genome? And so the reason that in my group, we frame this thing as basically a collective intelligence problem is that individual cells are extremely competent. So here you see one cell. This is a single cell organism. This is called the lacrimaria. And there is no brain, there's no nervous system, there are no stem cells, there's no cell-to-cell communication, just one cell. handling all of its local goals. So it's handling its physiological goals, anatomical control, metabolic, behavioral. Everything is handled by this one cell in real time. Okay, and so the amazing thing is that these cells, which are extremely competent in their local, very small sort of environment, when they work together to make a metazoan body, they can work on much bigger goals. So what you see is an inflation not only of body size, but actually of their ability to pursue states in various kinds of problem spaces, including anatomical amorphous space, physiological space, and so on. So a single cell can give rise to a collection of cells that self-assembles into some incredibly complex morphology. And we know that simply understanding stem cell biology is not going to be enough, because here we have a teratoma, and this thing might have hair and teeth and bone and muscle and skin. And so the work of the stem cells has proceeded fine. You have all your derivatives, you have your various tissues. What you don't have is this three-dimensional structure. So we need to understand how this works. And in a standard developmental biology, the story that we're told is a kind of a feed-forward open loop system, which is very much based around emergence. So the idea is that there are gene regulatory networks, so genes turn each other on and off. Some of these genes are effector code for effector proteins, so they are sticky or they diffuse or they exert force or something like this. And then there's this physical process where all of these things interact with each other. And then through things that are studied by the science of complexity and so on, out come these amazingly complex results. So like the salamander here. So this story is certainly true in the sense that these things all happen, but it's incomplete, I think, and it has a fundamental difficulty, which is this inverse problem. The idea is that if we're committed to this feed-forward emergence story, then making changes here require us to exert our interventions down here at the for example, at the genetic level. And that means that we have to try to invert this process of emergence, which is fundamentally insolvable. So, right, so most of these inverse problems are just too difficult to solve. How do you know what genes to tweak to make desired changes at the three-dimensional anatomy level? So the current state of affairs is basically this. We're very good at things like this, at figuring out the lowest hardware level of which genes and proteins interact with which other genes and proteins. But what we'd really like to understand are things like this: the ability of planaria to regenerate from pieces, the shape of the hand and the shape of the foot, and why they're different, and what you would have to do to get these specific shapes back. Okay, it's very not obvious what you would do at this level. So the amazing thing is that some animals are very good at this, and their bodies show remarkable plasticity in this kind of collective activity of the cells. So for example, here, this animal is a salamander. It regenerates its limbs, its eyes, its jaws, its portions of the brain and the heart. And so if they're amputated, they will grow back. And what's cool is that not only is this process incredibly flexible, meaning if you amputate at the shoulder, you grow the whole thing. If you amputate at the wrist, you start here and you just grow the parts you need, but then it stops. And that's the most remarkable part of all of this. And so lots of people work on trying to kickstart regeneration, but actually, how does it know when to stop? Because when it stops is when it has built a correct salamander arm. That's when it stops. How does the system know when it's built a correct salamander arm? Now, it's important to note that this is not just some weird quirk of salamanders. So the human mammals can do some of this. The human liver is highly regenerative. That's been known for a long time. Unclear to me how the ancient Greeks knew that, but they clearly did. The human liver is regenerative. Deer regenerate huge amounts of bone, up to 1.5 centimeters of new bone per day. Okay, every day, over 1 centimeter of bone growth. So bone, vasculature, innervation, skin, and even human children can regrow fingertips. Usually it stops at a particular age, but if you just keep it clean, the amputation will give rise to a cosmetically perfect finger. So the champions of this process are these guys. These are planaria. These are flatworms. They have a true brain, central nervous system, the same neurotransmitters that you and I have. And the amazing thing about them is that you can cut them into lots of pieces. The record is 275. And every piece will regrow exactly what's missing, no more, no less, to give you a perfect tiny little worm. While the new stuff is growing, the remaining tissue is actually shrinking so that they will as quickly as possible get to correct proportionality. And the other thing about them is they're immortal. There's no such thing as an old planarian. So if you're interested in aging or these ideas that the things inevitably wind down and accumulate errors and so on, planaria are telling us that that is not absolutely required. Here's a life form that's basically immortal. And the thing to understand about all of this is that we really are not, despite all the amazing progress in molecular genetics, we're really not very far along in understanding how these cellular collectives make decisions. And one of the easiest ways to see that is to just think about chimeric organisms. So here's a simple example, which we're making in my lab right now. Here's an axolotl larva. Axolotl larvae have legs. Here's a frog larvae, a tadpole. Tadpoles do not have legs. And one can ask a simple question. If I combine early axolotl tissue with early frog tissue, right? So in early embryogenesis, I make a chimeric embryo. They're perfectly healthy. We call them frogolotls. We have the genomes. We have the axolotl genome. We have the frog genome. And now we ask a simple question: do frogolotls have legs, right? And even though we have all this information, we actually have no idea how to predict in advance whether frogolotls will have legs, and if so, will they be made entirely of axolotl cells or both types of cells, or what? So these are the kinds of things that we would actually like to understand. And the one thing that's very important as part of this process is specifically the algorithm that enables robustness and the handling of novelty. And I'll show you a simple example of that. So we discovered this a few years ago. So here is a tadpole, and it has eyes here, it has nostrils and a mouth down here. All of these things have to move around in order to get to a frog face. Okay, so during metamorphosis, the face has to deform. The jaws have to come out, the eyes have to move forward, everything has to move. So it was thought that somehow what the genome did was to give each piece of the face a particular direction and amount of movement. And that way standard tadpoles become standard frogs. What we did was we created what we call Picasso tadpoles. So everything's in the wrong position. The eyes are on top of the head, the mouth is off to the side, everything is mixed up. I'll show you in a minute how we do it. But the amazing thing is that these animals become pretty much normal frogs, because all of these different organs move around through unnatural paths, and sometimes they go too far and actually have to back up, but everything moves around until it gets to a correct frog face, and then the remodeling stops. So in fact, what the genetics does specify is not a bunch of hardwired rearrangements, but a system that executes a really flexible error minimization scheme. It's able to start off at incorrect or abnormal positions and still get to where it needs to be. This parenthetically matches William James's definition of intelligence, which is the ability to reach the same outcome despite perturbations and starting from novel starting configurations. So how does this system know what a correct face is, and how do we get there? So we've been thinking about this problem, and we would like to. My background is computer science, and so to me, all of this looks like a problem of information processing at different levels. And what I would love to know is, could we go beyond the hardware and operating at this level and ask about the algorithms? How are these decisions being made? What do the cellular collectives measure? What are there modules or subroutines? How are these global patterning goals specified and stored? And in particular, how reprogrammable is any piece of biological hardware? So in computer science, there was this really interesting journey that we took from this is what programming looked like in the 1940s and 50s. In order to get the computer to do something different, you physically had to rewire it. You had to rearrange the hardware. You had to move the wires. But what computer scientists realized is that if your hardware is good enough, and I'm going to argue that biological hardware is definitely good enough, what you can do is something really interesting. You can reprogram the machine without touching the hardware, basically by giving it stimuli or experiences, basically inputs. And that will allow you to program in a much higher level language where you're not drowned in the details of the hardware or even the machine language, but you're actually able to take advantage of some really high level types of programming strategies to get very complex kinds of outcomes. And I'm certainly not arguing that living things are computers in the sense that of course, they're not built to the same architectures that our current computers are. But this idea of reprogrammable hardware and of controlling a system by inputs and stimuli as opposed to physical rewiring is very powerful.